Let's try to get your attention first thing. So you have 3 chapters on the test that we're working on for next Monday. And articulations is what we did yesterday, the joint classification, which joints are which type. And a lot of those questions, when you look in Canvas, what you're going to see is that you have a joint table that you can print. I was talking to Bettina and Robin this morning about making your own study tools, make your own. And the joint classification table is an example of one that is a format that I found works well for me, which is make a list of all the joints and then make a list of everything you have to know about each one. So your rows are each joint and your columns are what you have to know. What's their structural class? Is it fibrous, cartilaginous, or synovial? What's the functional class? Is it synarthrotic, amphiarthrotic, diarthrotic? For the synovial ones, you have to know if they're ball and socket or gliding or hinge or pivot or whatever. So I've got the bare bones of that table set up for you in Canvas, and you can just take that and run with it, print it, make your own, make flashcards instead, whatever you want to do, but at least it kind of organizes the information in a way that should be useful to you so that it's not all just kind of jumbled in your head. I think the hardest part of studying this much information is keeping it organized, right? So you all have a junk drawer in your kitchen, right? Oh, you should see mine. About once a year I reorganize it and I find, pieces of trash in there. Everybody in my family takes the scissors out of that drawer, cuts the tops off of bags, and puts the cut off top in the drawer. What is it with that? I don't know. It's because it's so full of junk already that it's just a mess. Anyway, if we need something out of that drawer, what do we have to do? If I need something that's in the bottom of that drawer, I have to pull everything out. And then I'm so frustrated I just throw it all back in there. So your brain should not work that way. Do not let your brain be a junk drawer. Okay? Make sure everything's nice and neat and orderly and in separate mental folders or filing cabinets so that if you need to know an axial bone, then you're sorting through all your axial bones. And you're not throwing arm bones and foot bones in there, right? It's not all just jumbled up. Keep it organized. It'll really help. So I'm going to try to work through muscle tissue organization with you today and try to get that organizational scheme first so that you know where to file the information when you get it. Before we do that, I want to point out to you that there's a homework assignment in Connect for this chapter. As there is for every chapter, this one's special. And there's another one like this that's special. In fact, there was a third one that I forgot to mention. The one on Chapter 4 was on membrane transport. And the thing about these homeworks is that they pull in, it's not just videos or animations in the book, it's interactives. And you have 3 of these in each semester. And the 3 you have for this semester were from the cell chapter, membrane transport. Do y'all remember looking at that? Okay. So the one for Chapter 10, this homework assignment only has 4 questions. That's it. Yay, that sounds great. But each question has multiple parts to it. So, but even if you don't like doing the homeworks, you'd rather just do the Learn Smarts, please do these because I put this homework in there specifically to give you easy access to those. They call them the integration concept overviews. And you have access to them in your book, but it's just kind of hard to figure out how to get there. So I went to my book, and I tried to figure it out. And even with me knowing what I was looking for, I still had trouble finding it. So the easiest way for you all to get to this is through the homework. So this is a view of the first homework problem. And you probably can't see that little teeny tiny screen, but up at the top here, you have only 4 questions, 1, 2, 3, and 4. You need to do all 4. Each question has 4 parts, and you can see the different parts. Here in green, you see the button highlighted for introduction, and then there's a strategy case. There's a few of those. There's a click and drag, and then another strategy case. So does everybody see how to navigate those? Okay, so when you get to question 2, you'll see there's a presentation. And that presentation took me a while to load. I mean, it came up first as a blank screen, and it took a couple of minutes. So give it time. But that is your
Skeletal muscle
interactive.
contraction depends upon the excitation of motor neurons that travel from the spinal cord to the muscle.
Okay, and then up at the top, you see blinking in purple a little
The
forward
nerve
arrow.
impulse or action potential from the motor neuron is relayed to the muscle fiber at the neuromuscular junction via the neurotransmitter acetylcholine.
And down at the bottom, you see it tells you what to do. Click and drag to zoom. So you've got a little blue arrow there, which lets you into the next part. So it's not just clicking the next arrow. You actually have to do stuff on several of the screens. Okay? So take the time to go through the Connect homework for Chapter 10. All right? That's going to give you the sort of animated tutorials that you need. You will also find that there's an APR for that chapter. Have y'all been doing the APRs? Some of you been doing them? Okay, so out of APR, what I did was I picked all the video tutorials, and that's what I threw in the APR homework. So even though you don't get a grade for that, it's really just a way for me to just kind of zero in for you so you know where the stuff I think is most helpful can be found. So go through the APR homework for these chapters as well, so that if you forget what I told you or you just need to hear it again, they do a better job than I do of explaining that physiology. And if you took intro, did you have the CD that came with your intro book? Do they still have the CDs? I have some of those. That technology is so old that a lot of computers won't play it anymore. But if you can get to that CD and it works on your computer, it really is very helpful. And if you want one of those and you have a computer that you can make play old, it's like old Flash and Macromedia stuff, Shockwave. So if you want one of those, I'll give you one because we have boxes of them and the technology is getting more and more out of date. We used to play them in the labs and they finally told us they won't go back and put retro software on our lab computers anymore. So the academic part of it is fabulous. It's just that they never updated the software to work with new browsers. So that being said, I think you'd be happier with that even than what your own book has. Okay, so use that if you think it'll help you, but definitely do that homework. Let's see if I can get this where I want it. Okay, so just a reminder that muscle is one of the 4 kinds of tissues. And when we did tissues, which was, just last week, we said at the time that we were going to defer muscle tissue till we got to muscles. Well, here we are. And what that means to you is that you have to go back to the tissues chapter and get all those details about muscle tissue. Now, we just really did a couple of things for each one. We said that there's striated and non-striated, and there's voluntary and involuntary. Now you have to get all the details. The one I'm least worried about is cardiac muscle, because guess what we're going to do next semester? You are, probably not with me unless you take it in the fall. But you'll be doing cardiac muscle histology when you do the heart. So for now, just the bare bones of cardiac, not a lot of details, but you'll see what's in the histology chapter. That was chapter 5, right? And this chapter is just really a minimum on cardiac muscle. So you need to know what there is in those 2 chapters about all 3 kinds of muscle tissue. Okay so far? All right. Your book has 700 skeletal muscles in the muscular system. That's one of those little bits of trivial facts that is not very helpful. A lot of those are paired, right? Not all of them are paired, but enough of them are paired that you're really not, even if you learn them all, you wouldn't be learning 700 unique names because so many of them have a right and left. Did you have to learn them all? Do you have to? I say did as if you'd already done them. That lab's tomorrow. And you'll find when you get there that we only assign about 35 unique muscles. Okay, 35. We used to do 50, and what we found is that most students just gave up with that. They were so intimidated by learning 50 that it was just like, yep, I'll just flunk that one. No, that's not okay. You can't just flunk muscles. And of course, everything you learn in lab, I also put on your lecture test so that you feel like you have a vested interest in doing well. So what you're going to find for muscles, the same as for bones, is that I'll test you in lecture on the names of the muscles, their actions, that's what they do, what body parts they move. We learned about the body movements just yesterday. And what we call their origins and insertions, which are the points of attachments of the muscles. And we also ask you the antagonist. Now, antagonist is not something we give you a list of. So let me tell you what the word means, and we'll come back to this later. But the antagonist has the opposite action of a particular muscle. So the muscle that does a body movement, like your biceps brachii flexes your forearm, we call it the prime mover. So if the biceps brachii flexes the forearm, what's its antagonist going to do? It's going to extend it, does the opposite. So once you've learned the actions, then just knowing what the word antagonist means, you'll figure it out. Okay, it won't be hard. It's not a challenge. Learning the muscles is the real challenge. Okay, and there are a lot of complete body parts we just totally neglect. So when you get there, you'll realize we're really shortchanging you, only making you learn 35. And when I ask my colleagues across the country how many, that's one of the big questions we all ask each other. How many muscles do you make your classes learn? And we're way at the low end. I never admit. I just keep mum when they ask that. I'm not going to admit we only do 35 because it makes us look bad. Most places the low end is 50 and some are above 100 unique muscles. So I know we're kind of shortchanging you guys, but I've never had a student come back and say, you should have taught me that muscle because I didn't know it. You'll find that somewhere, and I think it's going to be in the online stuff, I add a muscle. I'm not going to tell you today what it is because I want you to go read the overview for this chapter. You're supposed to read those overviews. So go read this one particularly carefully because it adds a muscle to the list of what you have to know for lab. Okay, so be sure you use your online resources for both lecture and lab. And one of the things I just added yesterday to your integration lab, y'all remember that thing, don't you? That lab that we posted completely online, and it doesn't have much in it yet. It had some stuff on scan. Did that show up on your practical? It was supposed to. Don't tell me if it didn't. I don't want to know. It was supposed to. It will show up on the second practical, and along with the stuff on skin, there's stuff on surface anatomy. So I think it was just this morning, I finally added the list of which muscle landmarks you have to know on the surface anatomy. Those may show up on your test in lecture. I do put surface anatomy on the muscle test. So can you tell from someone with their skin on, can you tell which muscle bulge is which? Can you tell where the deltoid is? Okay, that kind of a thing. So I don't think it'll be hard. I just want you to be ready for it. Okay, so we are going to focus our attention first on just the skeletal muscle. And so everything you're going to do today and most of tomorrow is going to be just skeletal muscle. That's how much there is to learn. We'll finish up with a little bit on smooth muscle, and then we'll defer cardiac muscle pretty much completely until next semester with just, again, that assignment from Chapter 5 and from this chapter on whatever's in the book. So for skeletal muscle, we know that skeletal muscle attaches to two structures. It attaches to the skeleton, but it also attaches to skin. So for example, muscles of facial expression may attach just to soft tissues. They may not attach to any part of the bone at all. And actually in other animals, they're what we call dermal muscles, and humans don't really have a lot of dermal musculature. We have a little bit, but not a lot. Where are you going to look for dermal muscles? That's a trick question. I think you might look in the dermis. Okay, where would you look for the origin of a dermal bone? In the dermis. So dermal musculature likewise. Dermal muscles in other animals is more useful than it is in us. If you've ever observed a horse, shaking flies off its rear end? How does it do that? Yes, it uses the tail, but what else does it do? It kind of shimmies its skin, right? Yeah. Can you do that? I'm thinking not. Pretty sure not. So the only dermal muscles that I can think of that you need to know, there's one in your neck that attaches to the corners of your mouth, your lips, and then just kind of spreads out this very, very thin, flat muscle on both sides. There's one on the left and one on the right. And it's a dermal muscle, so it does not attach to bone at all. What do you think it does? If it runs this way, it attaches to the corner of the mouth. What do you think it does? No, swallowing involves the throat, the tongue, and other structures. This is just dermal, so it's just right up under the skin, and it attaches to the corners of your mouth. If it smiled, it would have to pull the corners of your mouth up, right? So it helps you grimace or frown. So it pulls them down. If it's below, it's going to pull down, right? Remember that muscle shorten forcibly. So a muscle will always pull a body part towards itself, okay? So if the muscle is below the mouth, then it has to pull the mouth down. Does that make sense to everybody? Questions about that? All right, you'll find the name of that muscle when you get to lab. We won't worry about it. But that's the only one I can think of. Okay, I just lied. So there's one that is on your forehead. It's called the frontalis. And it's attached through a sheath, a big broad sheath of connective tissue, to the one on the back of the head. What do you want to call that one? If this is the frontalis, what's the one on the back going to be? Occipitalis. And those two muscles, because they're connected to each other, can be antagonistic towards each other. So the occipitalis, because it's pulling towards itself, is going to pull your whole forehead back. So when you raise your whole forehead and you can feel the whole top of your head getting pulled, that's your occipitalis. On the other hand, the frontalis, the fibers run up and down, so when they contract, it kind of wrinkles your forehead. Really important, right? Yeah. Life-threatening not to be able to do that. And yet we find that a lot of the muscles are muscles of facial expression. How important is facial expression? It's very important. You get a lot of visual cues from facial expression. And if you ever had a patient that couldn't express themselves through changes in facial musculature, then you would know how important it is. For example, people with Parkinson's disease. Very often the muscles of the face are affected, and they're either in a constant grimace or they just look stone-faced. They never change their expression. And it's hard to evaluate someone's mood when they can't express themselves that way. On the other hand, if there are people who can't read facial expression, isn't that likewise kind of a disability? So people with autism spectrum disorder fall into that category. They simply don't respond to normal behavioral cues, and one of those is facial expression. Okay, and that's a learned response. Anybody with little kids knows. They have to learn, right? I remember telling my kids, "Can you tell when I'm mad at you? Can you tell?" No, they could not. They still tend to ignore it, but that's a different issue entirely. All right, so muscles of facial expression, crucial, but most of the movements, as you'll see, are going to be movements that are attached to some part of the skeleton, either for just anchoring, like the muscles of facial expression, a lot of those are anchored to the bone, or for actually moving the bone. And those are going to be the ones involved in body movement. So contracting the muscles that are attached to bones produces some kind of movement. And body movements, as well as maintenance of posture, are really kind of the same thing. So if you're able to stay upright in your chair, so if you just sat up, not leaning back against the chair, but sitting upright, what muscles are contracting to keep your torso upright? Can you tell which ones are contracting? If you lean back slowly, which muscles are contracting to prevent you from falling back? Can you feel that? That's abdominal muscles. Okay, so you're using abdominal muscles and muscles of the back, a lot of which run along the vertebral column. You're using them together. Aren't they antagonists to each other? So if you contract them both equally, that prevents movement. So likewise, if I hold my arm just straight out, which muscles am I contracting to hold it? the muscles of abduction, which would be mostly the deltoid, but also the pectoralis and latissimus. And I know y'all haven't learned the muscles yet, so don't panic over my apparent assumption that you know what they are. Okay? But if I lower it slowly, then what? What muscles am I contracting to lower it slowly? You're using the muscles that help draw it down, but you're also using gravity, right? So if I just let it drop, I'm just relaxing muscles, which means the muscles of abduction, the ones that lifted it up, are also contracting to prevent it from just falling down. Okay, so postural muscles and muscles of movement are pretty much the same muscles. It's just a matter of degree of contraction and the relative contraction of muscles and their antagonists. Okay? And a lot of that is controlled subconsciously, right? Have you ever fallen over when you were asleep? I had a student one day who actually literally fell out of the chair in my lecture. Yep. It's the only time that had ever happened, just fell right out of the chair. Most of us wake up before we get to that point. He was pretty soundly asleep, what can I tell you? So there are parts of the brain, and we'll learn some of those parts of the brain. Just next week we'll be doing that. So you'll see how the nervous system and the muscular system work together, but you just need to think of it as a functional whole, okay? The nerves work with the muscles. And then finally, we have protection and support, and again, a lot of that is using the same muscles. What does your rectus abdominis do for you? It not only flexes the torso, it also protects the soft organs in the abdomen. So the story of the death of Houdini, have y'all heard that one? Anybody? See the movie? Anything? Wasn't that long ago, was it? You can Google that if you want. But the story of the death of Houdini was that somebody punched him in the gut, and he wasn't ready for it, and it, I don't know if it ruptured his appendix or what, but it did enough soft tissue damage that he died after that. So what Houdini, one of the many things he was famous for was that you could smack him in the abdomen and you just had no ill effects at all. But what was he doing, of course? If somebody was about to hit you in the gut, what would you do? You'd clench it, right? So he had really strong abdominal muscles, and he would contract the muscles to protect the soft organs inside. And when this guy asked him if he could try it out, he just didn't give Houdini time to get ready before he kind of sucker punched him, I guess. And so he wasn't ready. So do your abdominal organs protect, or your abdominal muscles protect the soft organs of your abdomen? Absolutely. Okay, so there's a lot of that. All right, so that's kind of your introduction to the functions of skeletal muscle. And again, for the most part, they're going to have to contract to do their jobs. And in contracting, they are going to be, why are we doing this? We're just not talking. When they contract, they shorten forcibly, and so what you have to remember about that is that they're always going to pull, if they're going to do movement, they're going to pull things closer to themselves. All right, so another category of skeletal muscles is sphincters, and you need to know what a sphincter is. A sphincter is a circular muscle, and some of them are skeletal muscle and some are smooth muscle. We'll worry about the smooth muscle sphincters next semester. You have them in your throat, you have them in your urinary system, you have them in your large intestine. Those sphincters are smooth muscle, and we'll worry about them mostly with digestive and urinary system. But the muscle that encircles your eye is a sphincter muscle. It's a circular muscle. The muscle that encircles the opening of your mouth is a sphincter muscle. And those are skeletal muscle sphincters, which means you have voluntary control over them. But by definition, a sphincter is going to regulate the opening of some passageway and regulate what can go through that passageway. And in general, and I say this pretty specifically, in general, the default position of a sphincter is to be closed. Okay? So in general, sphincters stay closed until you tell them to open. And when the stuff is finished passing through, the sphincter should close right back up again. Okay, so what's the default position of your eyelids? Closed or open? If you get so sleepy you can't control it, what do? They close. Okay. Never mind that I've seen people, and it's kind of spooky when they sleep with their eye open. Why is that spooky? Because that's abnormal, okay? And your mouth should close, right? Okay, so that's the typical position of a sphincter. All right, and then finally we have this totally different category of function of smooth muscle, and that is in heat production. So when you're cold, you shiver, and that shivering is the production of heat by your skeletal muscle. What are they doing? They're, in effect, wasting ATP. We talked about that before. We talked about wasting ATP as a mechanism that thyroid hormone uses to just generate body heat and keep your internal temperature up. So that shivering is a way to maintain internal body temperature. Josh?
Would the shivering be different than the thyroid?
Yeah, it is different. The thyroid's effect is on the mitochondria. And mitochondria is a different mechanism than muscle shivering. Does that make sense? There's
Would both be activated together, I guess?
shivering is has its own reflex. So when you get cold enough, and generally it's not going to be just your skin temperature, it's going to be core body temperature, then that sets up a reflex to the hypothalamus of your brain, which controls body temperature, and that then through intermediaries sends messages out to the muscles that shiver. Do all your muscles shiver? There are just certain ones, right? Your eyelids don't shiver usually, do they? They might get a nervous tick, but they don't shiver. Okay, so shivering is a specific mechanism. There is another mechanism, since you asked. We also produce brown fat. Have you heard of brown fat? Has nothing to do with muscles. It has to do with regulating body temperature. Brown fat is brown because it has a lot of mitochondria in it. What do mitochondria do? They can leak ATP. So particularly in babies, babies don't shiver. Did you know that? Babies do not shiver. They have a hard time thermoregulating, but they have a higher proportion of their body is in the form of brown fat. And most of that brown fat's right between the shoulder blades. There's just a little pad of brown fat in babies. It keeps the heart and lungs at a more normal temperature. And for a long time we taught that adults really don't have any brown fat, not enough to worry about, until somebody figured out how to look for it properly, and lo and behold, we do have brown fat. So the next thing I learned after that was there are all kinds of marketing gimmicks on how to eat diets that will let you cheat and eat a diet that's going to somehow generate brown fat. Yeah, y'all know not to believe that stuff, right? Okay, just thinking. Okay, so that's kind of an overview of the functions. Here are the characteristics. We've already talked about these ones. What did it mean to be excitable? If you can excite a tissue, that means when you stimulate it, it's going to do something. Muscle tissue, when it's stimulated, contracts. And remember that contraction means it shortens with force. And that tension that is generated, the force or tension that is generated, can be used to move bones. And then conductivity, we'll come back to. Conductivity just means the ability to conduct an excitable impulse. So we're going to get into all that. And then, of course, I had added one to that. What did I add? What does it mean to be extensible? What was it? If able to extend, which in the term of a muscle means what? Lengthening. Okay, without tearing. I think that's the main thing. You can lengthen a muscle without tearing it. Okay, and it was on the next page, elasticity and extensibility. What was elasticity? That recoil, so that the muscles, once they're to their fullest length, don't sit there and droop. They will return to their original length. And we'll look at what causes all these characteristics to exist. All right, so we're going to look at the gross anatomy of a skeletal muscle, and we're going to see that a skeletal muscle as an organ is made up of multiple kinds of tissue. It's more than just muscle tissue. There's going to be some connective tissue in there. There's nerves in there. There are blood vessels in there, so that's organs inside of organs, just like we saw with the skin and the bones. But we need to get a handle mostly on the muscle tissue. And so we've got some terminology here that you need to be aware of, and I'm going to try to keep track of these for you. So let's just go ahead and get started. If I have an entire muscle, in a single muscle, I have bundles of fibers, and each bundle is called a fascicle. The fascicles are the bundles of muscle fibers. And so it's a bundle of fibers. And a fiber is a cell. It's not really a cell, it's just the muscular equivalent of a cell. So what was a fascicle? It's a bundle of fibers. We're going to come back to this, but try to keep track of it. So skeletal muscle is composed of thousands of muscle cells. What do we call the cells? Fibers. And we put them in bundles called fascicles. So we have fibers, connective tissue, blood vessel, and nerve, all bundled up. And I don't know why this thing won't go to the next slide when I ask it to, but apparently it doesn't want to do that. All right, so we're going to look at the connective tissue components. Let's jump ahead and look at the picture. I know I have a picture in here somewhere. There we go. All right, so let's see what we can do with this picture. The whole thing that you see is a cutaway of a muscle. So in the cross section of a muscle, I have, can you see the individual bundles? There's a bundle. What's a bundle? A fascicle. And what's inside of each bundle? Fibers. Okay? What separates one fiber from the fibers around it? A layer of connective tissue. What separates this bundle from all the bundles around it? Another layer of connective tissue. What separates this muscle from all the muscles that it may be around? Another layer of connective tissue. Okay, so I'm going to give you the terms, and then we're going to see parallels. So the covering of each cell is called the endomysium. The covering of each fascicle is called the perimysium. And the covering of the entire muscle is called the epimysium. Okay, can you keep them straight? What does endo tell you? On the inside. What does peri tell you? Around. What does epi tell you? Upon. So sitting on the outside of. Okay? So keep those straight, because what we're going to see is that depending on what structure we're talking about, we have epi, peri, and endo associated with a muscle, associated with a tendon, and even associated with a nerve. So I have for muscle, the word root is mysium. For tendon, it's tendinium. And for nerve, it's neurium. So tell me what the epimysium is, the epimysium. Epi? It's the covering of a muscle. What's the epineurium? The covering of a nerve. What's the epitendinium? The covering of a tendon. Okay? What is divided into bundles? Muscle, tendons, and nerves, all divided into bundles. What covers each bundle? The peri. Tendinium, perimysium, perineurium. Okay? What covers an individual nerve fiber, muscle fiber? That would be the endomysium and endoneurium. Are you with me on that? Okay. So if we look at where a muscle meets a nerve, then we'll see that at the end of the muscle fiber, here's just the endings of a couple of muscle fibers, that there's an overlap between that and the tendon. So what layer am I drawing now in gray if I'm right on the outside of a muscle fiber? On the outside of an individual fiber. Endomysium. And the endomysium is completely continuous with the endotendinium. Okay? Can everybody tell that on the left I have endomysium and on the right I have endotendinium? How do you tell? That's a dumb question, I hope, but just to make sure. Is there any muscle on the right side of the picture? No, so I must be in the tendon, right? What if I wanted to draw the perimysium? I would put perimysium around an individual bundle, right? And then the peritendinium would simply be a continuation of that structure. Okay? So likewise with the epimysium, it's around the whole muscle, and the epitendinium is a continuation of it within the tendon. So if I drew them all, and of course I'm not going to draw any more muscle fibers, but if I drew just the perimysia of all of the bundles, do you see that? And the endomysia into the endotendinium. That once you get into the tendon, there's a really continuous connection with the muscle. So how easy is it to separate a muscle from its tendon? You really can't. You're more likely to actually rip the muscle fibers than you are to rip the tendon or to separate the tendon from the muscle. The most fragile tissue here is the muscle tissue. Well, actually, it would be the blood vessels, but we're not even to them yet. Okay? On the other hand, where a tendon attaches to a bone, it's not quite that straightforward. So if we take the knee joint, here's the bone of the knee, side view, here's the patella, so here's the tibia. One of the muscles that you're going to learn in lab between tomorrow and the next day is the big quadriceps muscle, which has like 4 different parts, and all 4 of those parts are connected by a tendon, actually technically by a ligament, to the patella, and then the patellar tendon, that's the tendon, and then the patellar ligament attaches to the bone there. Okay? So everybody got sort of a 3-dimensional view of what I drew? All right, so where am I on you? What body part did I just draw? The anterior knee. Okay, so there's the kneecap. So you have these big muscles of the thigh. They attach by a tendon to the kneecap, which attaches by a ligament to the tibia. Okay, you'll remember that tomorrow for sure. All right? So where that point of attachment is the weakest is right there, where it attaches to the patella. So if you overstress that big quadriceps muscle on the front of your thigh, that's a really powerful muscle. It's technically 4 muscles, 3 of which are pretty big. So, and they're the ones that are going to do what? What's their action going to be? They're on the front of the thigh, so what are they going to do? They're going to extend the knee. So that's a powerful action for kicking. Okay? But when you jump and land on your feet, you can actually really stress that muscle. So I know of a case of a guy who was hiking in the Alps in the summertime. It's a great place to go, I'm just sure, I've only heard. But he was hiking and he was on his way home, and it's really high altitude, right? Which means when the sun goes down, it goes behind the mountains fairly quickly. So you go from like broad daylight with the sun almost directly overhead to very quickly it's pretty dark. And he was high enough up that he didn't want to be out after dark in the Alps. Sounded like a good idea to me. So he decided he would take a shortcut instead of following the road, because the road, like a lot of East Texas roads, goes around people's fields. So he decided he would shortcut through this field. So he jumped the fence into this meadow to shortcut across the field and save himself a little time. And only after he got about halfway through the field did he realize he was sharing that field with the bull, and that the bull didn't want to share his field. So he started running to outrun the bull. He did outrun the bull. He got to the fence, he leaped over the fence, he landed on his feet, and he popped his quadriceps tendon, just popped it. How would you know? How would you know what you had done? What would be the expletive deleted moment for you when you realized as soon as you landed that something was wrong? What would you notice? Okay, there's pain. There's definitely pain. Anything else? Possibly. Yeah, that's not what happened in his case. He was probably holding onto the fence at that point. What?
Did he hear it first?
He probably heard a snapping sound. I've heard people have told me that they've done this with the biceps and that you can hear it. What function would he lose? He can't extend at all. So he did it on the right side, and he couldn't extend it at all. He could still flex it. What did that muscle look like when he looked down and realized what he had done? That muscle's not going to sit there all stretched out. It's elastic, remember? So it just recoiled about halfway back up his thigh, so this big bulge about halfway up his thigh. And he was an MD, so he knew what he had done. So what did he have to do? I mean, sun's still going down. He still has to walk to the village, down the Alps. So this is what he did. He walked like this. He had to use his hands to manually extend that leg. Pressing himself, no doubt, the whole way. Yeah. So the tendon didn't tear loose from the muscle. What did it tear loose from? It tore loose from the bone because, first of all, your patella is not that big of a surface to attach to. But so if this is an extreme close-up of the patella, and the underside is kind of fitted to your bones, there's the outer surface, then where it attaches, the collagen fibers of the tendon do insert and fuse with the periosteum of the patella, and probably to some extent into the bone itself, but not nearly enough, apparently. And it's not that uncommon a sports injury to actually dislocate the patella. And when people do that, there is, of course, cartilage on the underside of the patella. There's cartilage here and there's cartilage here because it's coming in contact with the femur and the tibia. And that cartilage can get very rough on the surface if you abuse it. So very often, particularly in young athletes, teenage athletes, that they will ruin that cartilage. And generally speaking, they're faced with two choices. One is surgery, and the other is quit running. So if you were the mom of the kid that always came in second on the track team, what would you tell your kid? You're not going to get a track scholarship anyway, sweetie, so quit running. So that's what me and my kid did. She still likes to run. Her knee doesn't hurt anymore. She did not have to have surgery, so just keep all that in mind. Okay, so does everybody appreciate how tightly the muscle and the tendon are connected to each other, and those three layers, endo, peri, epi? Got them? All right, you'll also see arteries, nerves, and arteries, veins, and nerves here, and those are going to branch within. So you get one artery, one vein, and one nerve to a whole muscle. And then you get a branch off of that artery, vein, and nerve for each bundle. Each fascicle gets its own branch. And then within the fascicle, each muscle fiber gets its very own capillary and nerve ending, all to itself. And this is something that a lot of times is not just really pointed out to people. So let's make a bigger deal of it than the books usually do. So if this is a muscle fiber, and they are kind of cigar shaped, they're kind of rounded at the ends, then the blood supply for that muscle fiber, here's the artery branch coming in. So here's the artery that supplies it. That artery will give off a branch to this one, and another one, and another one. So the artery's giving off multiple branches, but the branch that supplies this specific muscle cell, muscle fiber, is going to do this. This is the capillary. And then the vein will drain off the other side. So there's the venous drainage. Is everybody okay with that? Looks kind of like a hot dog, especially at lunchtime. So tell me why the capillary does this wavy thing. What's it doing? It's more, actually, technically it's more length, but it's the right idea. It's folding to increase the total coverage of it. So remembering that the muscle fiber is elastic and that this is a picture of its resting length, what will that look like when the muscle fiber is fully extended? It's got to stretch out. And when it stretches out, what do you want your capillary to do? Stretch with it, not rip to shreds. So the capillary's resting length doesn't change. It just has to kind of accordion itself up to fit along the resting length of the muscle fiber, which is elastic. Does that make sense? Okay, so when you overstretch muscles, can you do that? Can you overstretch muscles? Yes, you can. Particularly if you exercise more vigorously only extremely occasionally, and you don't usually bother. So if you never do any kind of range of motion therapy or exercise, stretching exercises, whatever, then your muscle fibers will shorten as a response to that. And as they shorten, the capillary will adjust to the new resting length or the new full length. So then if you overextend the muscles compared to what they're used to, then the muscle fibers still have some elasticity, they'll still stretch, but the capillaries tend to get torn. So when do you feel that? Right away? Does it hurt right away? Probably not. So have you ever gone out and done more physical exercise than you were used to, and the next morning you could barely move? Does that happen to you? Yeah, well, that's what you did. So you'll get over it. Trust me, you will. Because what those capillaries will do then is they will heal, and when they do, they'll respond to the new normal, which is I better grow a little bit longer than I was used to. So stretching exercises, at least, particularly as you get older, stretching exercises do in fact reduce the likelihood that that's going to happen. But in general, any exercise you do that's more than what you're used to is going to have that effect. Those little capillaries are just like, I don't know what to do, I think I'll just rupture. Okay? You're gonna be sorry tomorrow. That's what the capillaries do. Now let's look at what the nerve endings do, because again, the nerves are going to come in, they come in alongside of, oops, I hit the wrong button. The nerves are going to come in alongside of the, I have to start over. They come in alongside of the artery. So here's my resting muscle fiber, and here is my nerve. And again, it's going to give off branches to supply different muscle fibers, but the one that goes to this muscle fiber in general is going to hit it smack in the middle. So there's my little nerve ending, right in the middle of the muscle fiber. And skeletal muscle fibers absolutely have to have that. If you cut the nerve that supplies those muscle fibers, what do you think is going to happen next? That nerve's cut. That muscle fiber is paralyzed, can no longer contract. Please do not practice this at home. Okay? So the term for a muscle that cannot contract, it has no muscle tone, it's completely flaccid. Flaccid or flaccid? Flaccid. Okay, questions about that? All right, so there's more than just this picture to what meets the eye in terms of the muscle as an organ. Let's go back through our list of slides and make sure we got everything. I think we skipped a couple of things. So we had our connective tissue components. We had epimysium, perimysium, endomysium. We got those, right? One thing I didn't mention about the endomysium is it doesn't just connect the muscle fiber to the tendon. It also insulates that muscle fiber from the neighboring muscle fibers. It prevents the electrical signal from short-circuiting between two different muscle fibers. And then, of course, we have the tendon. I gave you the term aponeurosis the other day. Does anybody remember? It's like a broad, flat tendon. And the term aponeurosis tells you it's upon the brain. That's what it is. So that big sheet of connective tissue on top of your cranium, that's the aponeurosis. But we use that same term more broadly to refer to other big flat sheets. It's basically a big flat tendon. Okay, there's a couple of other things. There's a deep fascia. We had already learned the superficial fascia, right? Where's the superficial fascia? That was on your test yesterday, wasn't it? Anybody want to volunteer that they remember what the superficial fascia is? It's the same thing as the hypodermis. It's the subcutaneous layer. Oh, yeah. So it was a skin-related structure. Remember that story about the junk drawer I was telling you? Yeah. File your superficial fascia with your skin. Okay? File your deep fascia with muscles. The deep fascia is external to the epimysium. So it's outside of that and not considered part of the muscle, but it does help separate that muscle from neighboring muscles. So, for example, in the anterior surface of your forearm, you have muscles that run side by side. You can see the tendons of some of them in your wrist. So you know you have individual muscles there. Yesterday we learned that there's a synovial membrane separating the tendons. What separates the muscles from each other is the deep fascia. So I can contract one of those muscles without affecting the neighboring muscles too much. Okay, that's what the deep fascia does. It separates one muscle from the neighboring muscle. Okay, so superficial fascia we did. And we talked about the blood vessels and the nerves, specifically the nerves that control muscles are called motor nerves, and the nerve cells are motor neurons. And we'll come back and talk about that some more later. And we talked about the fact skeletal muscle is voluntary. What did that mean? You have conscious control over it. Okay, so now we're going down to the microscopic level. And here it helps to have that organizational view. So let's go back to the idea that we had before of the fact that you had a muscle. What was the muscle divided into? Fascicles. And what made up the fascicles? Fibers. So now what we're going to do is go to the inside of a fiber and look at the microscopic anatomy. And when we get to the inside of the fiber, we're going to find that it is composed of filaments. And those filaments are likewise divided into bundles. So I have filaments, but the filaments are organized into bundles, and I don't want to confuse my labels by using the same label twice, so I'm going to call the bundles within a fiber the fibrils. Is this confusing enough for you? You have 4 words that start with the letter F. What a pain. So you have a fascicle, a fiber, a fibril, and a filament. And most of the time, just to make darn sure everybody knows what we're talking about, we call these myofibrils and myofilaments. What does myo mean? We use it to refer to muscles. It literally means mouse, but never mind, so does muscle. So the genus name of the mouse is mus. And anytime you put CLE at the end of a word, it means little. So what does the term muscle literally mean? Little mouse. Because to the Greeks who named it, muscles look like little mice jumping under the skin. I do not make this stuff up. Somebody else may have made it up. I don't know. But that's the story I heard. All right, so can you keep straight? Fascicle, fiber, fibril, and filament. Which ones are microscopic? Can you see muscle fibers with the unaided eye? No, they're cells. You need a microscope to see cells. Can you see fascicles with the unaided eye? Yeah, when you take meat and you shred it, that's what you're looking at. You're looking at the individual fascicles. Okay? All right, so we have some terms, and I'm sorry that some of these terms don't seem consistent. So we have the term sarco, which literally means flesh, and the term myo, which means muscle. And when you think of the term flesh, what do you usually think of? You think of skin, I bet. But flesh literally means the edible part of an animal, which for most of us would be not the skin, but the muscle. Okay? So you've heard of a sarcophagus before, right? What is a sarcophagus? They found King Tut's sarcophagus. It's something you put the coffin in, actually. It's usually an ornate cover for the coffin itself. But a sarcophagus is what you inter a dead body into. Are you with me on that? So why is it called a sarcophagus? Flesh eater. A macrophage is a big eating cell. Phagocytosis is cell eating. So sarcophagus is flesh eater. That's what the term literally translates as. Okay, so if you need to grow somebody out this week, now you know how to do it. Okay, so we use the word root sarco in addition to the word root myo to refer to muscles. I don't know why that is. It's just the way it is. So sarcoplasma would be the cytoplasm. Sarcoplasmic reticulum, what would that be? What does it sound like? It's SR for short. It's the same thing as ER, only highly specialized. What is ER inside of a cell? Endoplasmic reticulum. So the sarcoplasmic reticulum is the highly specialized ER, smooth ER of a muscle cell. Okay, so that's some examples of the word root, the use of the word root. Muscle fibers are not truly individual cells. Like osteoclasts, they're the fusion of lots of cells together. And like an osteoclast, all the nuclei persist. So a single skeletal muscle fiber may have 100 nuclei. It's 100 cells fused together. The individual cells are the myoblasts. Does that make sense to you? We learned osteoblast, fibroblast, chondroblast. The blast cell is the forming cell, the immature stage of the cell. So myoblasts are the embryonic precursors of the muscle fibers. And to form the mature muscle cells, they fuse into one giant muscle fiber. And some of the myoblasts will persist. I missed my picture somewhere. Yep, there it is. Some of the myoblasts do persist, and they call them satellite cells. Why would they call them satellite cells? Where are they? They're just kind of orbiting around the mature cells. So satellite cells are basically myoblasts that are in sort of a state of suspended animation. What are they waiting for? If you cut it when it heals, some of those satellite cells can actually wake back up and do some muscle healing. In adults, typically not enough to regenerate a lost function. But in other animals, we know that the myoblasts are fully functional. For example, if you startle a lizard of a certain variety, what will it do? Skinks are notorious for this. They lose their tails. Their tails will come off. If you try to catch them, even you will find them every once in a while in this room. And so we turn them loose in the greenhouse. They eat bugs, and so we like to have them around. They're little Mediterranean geckos, and when you see them, they might be only an inch long, and they're practically translucent. They're just sort of beige, and they look like have a cloaking device or something. They're almost invisible. So if you see one scurrying around, you can let me know. I'll try to catch it and put it back in the greenhouse because there's not much to eat in this room. And they're not native to this area, but somebody released them. When we first moved to Tyler, that was in the 1980s, they were known in the Dallas area and not in East Texas at all. So us being biologists, my husband found some down in I can't remember where he was, somewhere in the Dallas area. So he brought a couple of them home and we turned them loose. And we weren't the only ones that did that. So now on my front porch, I have so many of these things, and they get this big. And they eat the bugs. They're very pale in color, yeah. Like practically just a pale beige with maybe a little bit of a marking on them. And they come out at night and they eat bugs. So don't hurt them. But if you try to catch them and you do a bad job of it, which I've done several times, their tail pinches off. Just pinches right off. This is a defensive mechanism for the lizard. What does the tail do? It sits there having muscle contractions, and so it twitches, and that distracts the attention of whatever predator was trying to catch the lizard. The lizard runs the other way and lives to grow a new tail, which is why we're talking about this. So they can regenerate muscle and bone and nerve and skin. And we would love to know how they do that and harness that for human regeneration, but we haven't got that far yet. Anyway, in them, but not in us, the muscle can regenerate. But that's the story of the myoblast. So if I look at these huge muscle fibers, then what I see is that they have multiple nuclei. Each nucleus is responsible for the protein synthesis in its own little part of the cell, and also for maintenance of the other structures. So some of the other structures are parts of the membrane, so the membrane components. And again, I'm trying to clean out your junk drawer ahead of time. Make a folder for your muscle fibers, and in that folder, I want you to divide up those things that are part of the membrane. The outside membrane, the plasma membrane, is called the sarcolemma. And it has invaginations. What does that mean, an invagination? It folds inward. So if I looked at a sectional view of the membrane, here's the membrane, what I would find is that there are little openings, and in those openings there are these tube-like extensions called invaginations. These are the T-tubules, and we'll see why they're called transverse tubules when we get oriented. Okay? So far? We'll come back to what they do. That's too much information now. So what about sarcoplasmic reticulum? What is it? It's the smooth ER of the cell, so it's membrane. And where the sarcoplasmic reticulum comes close to the T-tubules, you have these structures called triads. How many parts are there to a triad? 3. So you have SR on each side and T-tubule in the middle. We're going to see a better picture of that in a minute. Right now, we're just associating these things with membranous components. Are you with me on that idea? So can you name 1, 2, 3 different membrane components of a muscle fiber? There's 2. Went too far. Sarcolemma. What was the sarcolemma? Just the plasma membrane of the cell. Okay. What are the T-tubules? Little extensions into the cell from the sarcolemma. What was our third membrane component? Sarcoplasmic reticulum. What's our abbreviation for that? SR. Okay so far? Now, your book is going to get next into the function of these, and I'm not ready to do that. I apologize, first of all, because I should have rearranged these slides. But what I want to do next is talk about the rest of the structure of the muscle fiber before we get too bogged down in what they do. So you remember our hierarchy. We had a whole muscle. It was divided into bundles called fascicles, which were made up of individual cellular components called fibers. Each fiber is divided into myofibrils. Each myofibril is composed of myofilaments. So we have the muscle fibers, that's the cell. The myofibrils, that's the bundles inside of the cell. And the myofilaments, which are the proteins that make up the bundles. Okay? Yes? No? Maybe? Okay, so are any of these things membrane? Myofibrils or myofilaments? No, these are non-membranous. Okay, so we had 3 things that were membrane. What were they? Sarcolemma, T-tubules, SR, sarcoplasmic reticulum. These are non-membranous. These are protein filaments. And they're going to be arranged in a particular order and organization that's going to be really important for you to understand. So let's go ahead and get started on those. All right, so here's your picture. When I look at the structure of the protein filaments of a muscle fiber, what I see is that I have 2 kinds of filaments. And we just call them thick and thin myofilaments. But the thick and thin myofilaments overlap each other in a way that creates a pattern. And so you need to understand that pattern. And the pattern shows up here in the unit that we call a sarcomere. So remember, sarco is just the word root we use to refer to muscle. And mere, M-E-R, E refers to body. So it's the unit of construction of the myofibrils. Okay? So far? Maybe? How many filaments have I described? 2 of them. All right? So I think what we're going to do, y'all are definitely starting to zone out, and I can't say that I blame you. So we're going to try to draw those. All right, so in your groups, try to draw that. Use your book as a reference. What I want is not that much. I want 2 sarcomeres, exactly 2. No more than 2 sarcomeres. Use your book, use your lab manual, use your mobile device, use the picture on the screen, call me over for help. I want 6 thin filaments and 5 thick filaments in every group. 6 thin filaments and 5 thin, 5 thick in every group, and I want 2 sarcomeres. And that's all the information I'm going to give you to start with, because I want you to try to figure it out. Okay, so does everybody see 3 Z lines on the screen? Z discs? We used to call them Z lines. It's hard to change. Okay, so which one's attached to the Z's, the thins or the thicks? Thins. And do they run all the way from one Z line to the next Z line? No. Each Z disc has thin filaments going in both directions. Mine are crooked. Technically, they should all be exactly the same final length. And of course, if I were doing this following my own directions, how many would I have exactly? 6 thins and 5 thicks, right? That's what I told you. I'm not counting right now. All right, so your thins should look something like that. Where is the Z disc? Right smack in the middle of the thins. Are you with me on that? Okay, so then to make my thicks, where are they going to go? In between the thins. Do they touch the thins? Not really. So they kind of sit in between. And again, they should all be the same length, lined up with each other. Is this what your drawing looks like? Sort of, kind of? Josh?
On the diagram, like it is indents, like it shows connecting coming out of that and connecting to the thick.
Yeah, so there are these other proteins that are involved. So I am, at this point, oversimplifying things, technically. What happened there? So you're saying the thicks are attached, or the thins?
Like where it like indents. Yeah. Yeah, there's some things in there, not that but too much.
Yeah, that's beyond what I care about, so I didn't pay any attention to that. Okay? All right, the degree of overlap is not a static thing, it changes. So if you look up here, this is my handy high-tech demo. These fingers are thin, these fingers are thick. You just have to imagine that. All right, do they overlap with each other? They have to. What would happen if they didn't? They have to grab, the myosin has little heads that has to grab onto the actin. If there's no overlap, there's nothing they can't grab. That happens if you overextend a muscle. That's really hard to do. So, in fact, it's almost impossible. The only time I've ever heard of anybody overextending a muscle is when they were trying to lift too much weight or they broke something. They actually broke their elbow joint. Can you overextend a muscle? Under ordinary design constraints, no, it's just not going to happen. But watch again what happens when the muscle contracts. The thin and the thick overlap more. They slide together. And that sliding together increases not just the degree of overlap, but a number of myosin heads that can grab on and hold. Okay? So if a muscle's already as short as it can be, it can't shorten anymore. It can't generate any more force. So if you have a muscle constrained so that it can't shorten, then it can't generate any force. It can get tense, but it can't shorten anymore if it's already as short as it gets. Okay? So this is where the action is. This is not the whole story, but this is where the action is in what the myofilaments are doing. So we'll go back to the picture that your book has. This is the one that you were objecting to my rendition of. Just a little, artistic license there. So there are some other things going on in here, but let's just focus on the things you need to know. So the Z-discs are important because those are the points of reference. So we need those, Z-discs. The thick and thin filaments, we need those. And there's some other structures in here that I do expect you to learn. And of course, your definitive source is what's in the online materials. But we have these things called the I bands and the A bands. And I want you to tell me what the defining characteristic of the A band is. You have the little dashed lines here showing you where the boundary of the A and the I are. What's in the A that defines it? The thick filament. So does everybody see that the A band is as wide as the thick filaments are long? So the thick filaments are what define the A band. And by defining the A band, by necessity, they also define the I band. What's not in the I band? The thin filament, or excuse me, the thick filament, not in the I band. Okay, so what is in the I band? Thin. Is the I band do you have thin filaments in the A band? Yes. So we have these different zones. So in the I band, we have thin only plus the Z, right? In the A band, we have this area where thin and thick overlap. We call that the zone of overlap. That one's tough. So there's the zone of, oh, sorry, messed that one up. Where's the zone of overlap on the other side? Zone of overlap from here to here. Does everybody see that? Okay, so what's left in the middle of the A band? That's the H zone. And what's in the middle of the H zone? The M line. Are you with me on all that? It's just a bunch of alphabet soup, you guys. And you just got to remember what's with what. I could tell you what they mean. I means isotropic. A means anisotropic. H means bright, only it's in German, Hella, which is bright. I can't remember what the M stands for. Okay, but you have to know the parts of a sarcomere. And you have to be able to label the Z line, the H zone, the I band, the A band, the M line. And you have to tell me where the zones of overlap are because that's where the action is. So again, looking at my handy high-tech demo, what happens when I slide them together? What happens to the zone of overlap? Doesn't it increase? So what happens to the H zone, which is thick only, and the I band, which is thin only, if I increase the amount of overlap? I have less area that there isn't overlap. So now what I want you to do is draw one sarcomere completely contracted. One sarcomere completely contracted. So in the completely contracted state, what do the thin filaments on the left side do compared to the thin filaments on the right side? You need some Z lines in here. Z discs. So everybody see how they just butt up against each other. The thick filaments are completely up against the Z lines or as close to them as they can get. Thin filaments from the two sides may completely overlap. And if you looked at the adjacent thick filaments from the adjacent sarcomeres, they'd be right smack up against them. So what happened to the H zone and the I band? Where are they? They're basically disappeared. So you just have thick filaments butted up against Z lines. Can everybody picture that okay? So if we go back then to our view that shows one sarcomere next to another sarcomere, then we're saying that the area of the I bands, here's an I band, right? There's an I band, there's an I band. That they're just basically gone, right? And it's just the thicks are all that's visible. So I need y'all to come up here. Let's see, we're just going to go kind of make a semicircle. Everybody gets to participate since the class isn't that big. Just make a semicircle up here. Okay, what I need you to do is use your arms as thick and thin filaments. So that does mean you have to spread out. So I want you overlapping just at the hands. Don't do anything that's going to make your neighbor slack you. Just overlapping at the hands. Keep coming this way. That way, that way, that way. Scoot, scoot, scoot. Okay, stop right about there. I think that's probably going to do it. All right, overlapping hand to hand, arms outstretched. You have no elbows. Arms outstretched. And overlapping hand to hand, which means y'all need to start scooting back the other way. We're going to make just a big happy circle, it looks like. Yeah. Come on, just a little bit more this way. All right, far enough. That's far enough. Stop right there. Overlapping, overlapping, just hand to hand. Okay, so now what are you going to do? You're thin, okay? You're thick, thin, thin, thick, thin, thick, thin, thick, thick.
I'm big.
Thick, thin. All right, if you're thick, you have myosin heads, and you get to move your wrists. Still have no elbows. Rest your arms whenever you can. If you're thick, grab the hands of the thin filaments to your left and right. If you're thin, you're passive. All you get to do is stand there. If you're thick, this is the fun part. You have to pull toward yourself. And the only people that can truly move are the people that are ends. So pull toward yourself. As you pull, you're pulling that whole string of people. As you pull, you're pulling. So you're pulling likewise, you're getting pulled that way. Okay, when you're pulling that of your actin, the thin filaments. And pull again. Pull again. Pull again. Pull. Remember, we're going to go towards the middle. Okay? Because only the ends can truly move. Now, what you have to remember is that the guys at the ends, you and your free hands are actually attached to the plasma membrane. So you're literally pulling the plasma membrane towards you. What did we just do to the whole length?
Shortened it.
We shortened it considerably, didn't we? Did we? All right, thin guys, you have no elbows. Remember that?
No,
There you go.
So all we did was move you closer together without shortening the individual filaments, right? Hold your pose. 1 second. See if I can do this again. Are you holding it? Okay, you can go sit down.
they were pulling you too far. I was trying to make it even.
I should probably get a signed release from you guys, but okay, so what you got from that, I hope, was the sense of the tension that's on each sarcomere. Each sarcomere individually is utterly connected to its neighboring sarcomeres. And so when you pull at all, they're all pulling on their neighbors left and right. What's happening to the whole thing? It's shortening internally, but the only pull is at the ends, okay? So that whole muscle fiber is going to get shortened. If it was this long, the whole thing is going to get shortened. Now, as it shortens, doesn't it also thicken? Not very well drawn. Don't your muscles bulge when they shorten? Because all that stuff has to go somewhere. If I shorten it, it's got to get wider. Are you with me on the concept of all that? Okay, so everybody has a good sense of how the filaments slide. Now, what we haven't figured out yet is what makes them slide, what makes them relax, what are the triggers. And that's getting down, that's not even microscopic, that's molecular biology, understanding the structure of these proteins. And there's lots of proteins in here that, honestly, it's just gotten to the point where they keep finding new ones all the time, and they don't inform the story. Have you ever had someone that told a story with too much detail? Like they're going to tell you exactly what they ate for lunch when really the story is about what happened after lunch. You know what I'm talking about? That's the way I feel about muscle physiology. It used to be just, you got these thin guys, you got these thick guys, you have these Z discs that you can watch, and you can actually watch them under the microscope. That's all an important part of the story. But when you get in here with these things that they found, that you've got this connectin stuff. Connectin's absolutely fascinating to me. You've got stuff called titin, which is really cute. You've got dystrophin, which is just another protein. Fine, fine, fine. If you're a molecular biologist, you know, have fun with that. But for you to understand the basic physiology, you don't need any of that. Okay? So I am not requiring you to learn all the molecular detail, just enough to understand the basic story. And that's it. So when we look at this, what we're going to look at is the fact that you have these myosin heads. What do they do? Here's some myosin heads. What do? They grab the actin. That's important. How do they do that? How do you regulate it? How do you make them let go when you're ready? Those are questions that you should know the answers to. And then for the actin, what part is being grabbed and how do you stop it from being grabbed when you don't want it? So what you'll see in the picture in between the myosin and actin, and in sort of a blue color here, is a little regulatory cluster of proteins. And those you kind of need to know the story of. So I think we have enough time to tell that story. And so we'll go through that, and then we'll probably call it a day. All right, let me get rid of all that. Okay, so what did we have? We had a protein in the thick filament called myosin. And myosin has these things on the top called heads. And the heads actually attach to the main protein in the thin filament, which is called actin. And actin attaches to the heads at a little region called the active site, which you're familiar with that term from studying enzymes, right? Okay, so if I were going to draw that, my head on my myosin, and it's actually kind of a double head, and then here sits my actin, and here's its little active site. So the head is going to attach and it's going to pull, and it's going to pull towards the middle of the sarcomere. So what I need in addition to this is I need something to regulate that. And so I have this little regulatory guy that covers the active site on the actin until I'm ready for contraction to occur. And then I uncover the active site and it just starts moving. So this regulatory structure is a combination of two proteins. One is called troponin, which means the activator, the twister. And the other one is called tropomyosin. And between the two of them, troponin and tropomyosin have to do a couple of different things. First, they have to grab and cover the actin. But then they have to be able to twist out of the way, which means they have to know when it's time to twist out of the way, which means they have to have an active site for something to activate them. And I'm going to go ahead and give away the plot. We're going to use calcium to activate. Okay, so we're going to throw some calcium ions loose in this muscle fiber, and when we do, the calcium binds to the troponin, and the troponin pulls on the tropomyosin, which twists the whole thing off the active site of the actin. And once the active site on the actins are exposed, the myosin heads start grabbing and pulling. Can everybody visualize that series of events? What came first? Calcium. Throw that calcium in there. What does it do? It binds to the troponin. What does the troponin do? It twists the tropomyosin out of the way so that it uncovers the active site. Then what? Then the myosin heads can attach to the exposed active site on the actin and start pulling. And as long as the calcium's loose, then the troponin tropomyosin stays out of the way. Myosin attaches, pulls, let go, attach, pull, let go, attach, pull, let go. It pulls as far as it can until the calcium is removed. Okay, so here's the big picture for this part of the story. If there's calcium loose in a muscle, what's it going to do? It's going to contract. It's going to contract until you get that calcium out of there. Okay, you put calcium in a muscle, it's going to contract, and it's going to stay that way.
Then does it have like a resting stage?
Yeah, if it's working right, you don't leave the calcium there. You're going to get rid of it. You're going to pull it out of the cytoplasm of the cell and let the muscle rest. But if you can't do that, if you put calcium in that muscle and it's too much for the cell to get rid of, what's that muscle cell going to do? It gets into a state of rigid paralysis. It contracts and it stays that way. So if you have too much calcium in the body, you can actually have muscle contractions, uncontrolled muscle contractions. Now, we already know what hormone controls calcium. Do you remember we did that? It was just last week. Everything was just last week. You had these calcium regulators. Parathyroid hormone raises the calcium, and calcitonin lowers it. And calcitriol, our other hormone, helps parathyroid. Parathyroid is the acute hormone, that's the short-acting one. Calcitriol is the chronic one, the slow-acting one. Are you with me on that picture? There are people who have parathyroid hormone tumors that hypersecrete PTH. Anybody want to guess what the effect of that's going to be on the muscles? Talk about it at your tables and write it down. What is the effect of hypersecretion of PTH? What do you see in this picture? Hyperparathyroidism affects the extremities before it does the center of the body. And so what we see typically is a condition called tetany. What is tetany? Tetany, like tetanus, means contraction. So tetany is the muscle spasms that you get with hyperparathyroidism. Why do you get muscle spasms? What you get is flexure of the wrist and fingers, and you get what we call a spasm of the foot. It's called carpopedal spasm. Why is it called that? Carpo for wrist and pedal for foot. Carpopedal spasm is seen with classic hyperparathyroidism. Why are we seeing that? What do you have too much of in the blood with hyperparathyroidism? Calcium, and the calcium level is so high that the muscle cells can't keep it out. And when muscle cells take in calcium, what do? They contract, and they stay that way because they can't get rid of the calcium in this case.
When you die, doesn't your body go into rigor mortis?
Yeah, you want to talk about rigor mortis? That's a different problem, but
Not if there's pictures involved.
no, I'm not going to show you pictures of rigor mortis, although I can tell you stories. No, we'll save it for later. That has to do with the way the myosin heads work. So the myosin heads require ATP to function. Oh man, they got all kinds of things. Nebulin, dystrophin, never mind all that. All right, let's get to where the action is. Since you brought it up. Not there, not there, not there. So we're kind of jumping ahead, but I'm okay with that. So if you're trying to keep up, this is figure 1011. And what you're seeing here is the steps involved in what we call cross-bridge formation, which is the attachment between the myosin and the actin. So myosin grabs the actin, holds onto it, doesn't just hold it, it pulls it, but then it has to let go before it can reach back and grab another actin. So if you look up here, I'll try to demonstrate the steps. My wrist is where the head has its hinge. Okay, so my head has to reach up, grab an actin, pull it, let it go, and then reach back. Okay, so in its resting position, it's kind of like
kind of like a bear trap. It's been set, it's ready. So as soon as the actin is available, it grabs it, it pulls it, but it doesn't just let it go. To let it go requires ATP. So I have to add an ATP before it will even let go of the actin, and then it will break down that ATP to reset itself. Okay? So it grabs the actin, it pulls, then you add ATP, then it lets go of the actin, then it breaks down the ATP and resets itself. Does everybody see the steps involved? So in rigor mortis, what happens is after you die, I don't know if you knew this or not or thought about it, but after you die, you could make an ATP. But you still have some stored. But because membranes and ions start to break down, membranes start to break down, ions start to diffuse, those membranes will discharge, and that lets calcium loose. Calcium starts leaking into the muscle cells from their intracellular storage. And so that calcium leaking out makes your myosin heads go crazy. They're pulling like crazy, using up the stored ATP, which will last not very long, a couple of seconds at most. So it's not generally all the muscle cells at once, so it's not like the body moves for 2 seconds and then quits, but one cell after another starts to tense up. And over a period of, depends on how warm the body is and all that, over a period of time, the muscles will tense up. And that's what happens in rigor mortis. It takes ATP to let go. You run out of ATP. So the myosin stays bound to the actin and the muscles stay tense until the body decomposes even more, and then the muscles start to break down. Does that make sense to everybody? So whatever the body's position at death, that's the position that tends to persist. So when one case I heard of, the guy tried to say grandma died peacefully in bed when the position of the body indicated she'd been stuffed in the closet. So you can't move, I mean, you can't lay them out. The muscles are rigid and they won't let go. So that's used for forensic analysis, of course. There's also a condition called lividity, which if you've ever watched those crime scene shows, does anybody know what lividity is? The blood tends to settle at the lowest point. So if Granny died peacefully in bed, then why is all the blood settled into her buttocks and her feet where she was stuffed into the closet? That guy didn't get away with it in that particular case. So again, I'm not trying to teach you guys how to get away with it. Really, really not. But it's important to just be aware of it. Okay. By tomorrow, it would be great if you had been through the homework for Chapter 10 and that you had learned about how calcium works, how the myosin and actin do their thing, and through how the nerve stimulates the muscle. We're going to go over that in class, and I'm a lot more compassionately understanding with my summer students because I know we have a really squashed schedule. So if you haven't done that yet, don't feel extraordinarily guilty and skip class. Feel guilty, but don't skip class. Okay? The more you've looked over it before you come to class tomorrow, the more you'll be able to absorb what we're talking about. In lab tomorrow, you're actually going to do a little bit of muscle physiology. So you're going to actually get to zap somebody in your class with electrical stimulation and make the muscles contract and study the effects. So we'll be talking about that in class tomorrow as well. Tomorrow's Wednesday. Yeah. We may actually get through with muscle tomorrow. And if we do, we'll get a head start on the nervous system on Thursday. Okay? Wait, what's today? It's Tuesday. Yeah, I haven't lost a day yet. Okay. All right. Further enough. All right, see you tomorrow.